Hello everyone, I'm Muskan from Sri Venkateshwara College of Engineering, and today I'm going to be presenting on the topic Analysis of Completely Randomized Design. Definition of Completely Randomized Design. CRD is a research experimental design where subjects or items are randomly assigned to different treatment groups. It ensures equal chance for each experimental unit to receive any treatment, minimizing bias and enhancing statistical validity. The randomization process in CRD. In CRD, randomization involves assigning experimental units or subjects to different treatment groups through a random process. This ensures that each unit has an equal and unbiased chance of receiving any specific treatment, reducing the influence of confounding variables and enhancing the validity of statistical analysis. Let's see the importance of CRD. It minimizes bias. CRD ensures unbiased allocation of experimental units to treatment groups through randomization, reducing the impact of potential confounding variables and producing more reliable results. Statistical validity. Randomization enhances the statistical validity of experimental findings by creating groups with comparable characteristics, allowing researchers to attribute observed effects to the treatment rather than pre existing differences. Assumption of independence. CRD assumes independence among experimental units, facilitating the application of statistical tests that rely on this assumption. This assumption is crucial for accurate hypothesis testing and result interpretation. The simplicity and practicality. CRD is straightforward to implement and practical for experiments with a single factor. It simplifies the design process and makes it easier to analyze and draw conclusions from the collected data. Generalizability. The random allocation of treatments in CRD contributes to the generalizability of study results, increasing the likelihood that findings can be applied to a broader population or context. Randomization control. CRD provides control over the randomization process, reducing the potential for systematic errors and ensuring that any differences observed between treatment groups are likely due to the treatment themselves. Let's see the key characteristics of CRD. Random assignment of treatments. In CRD, treatments are randomly assigned to experimental units or subjects. This randomization ensures that each unit has an equal chance of receiving any specific treatment, minimizing selection bias and creating comparable groups. Independence of experimental units. CRD assumes independence among experimental units, meaning that the response of one unit is not influenced by the response of another. This assumption is essential for the validity of statistical analysis and allows for the application of various statistical tests. Homogeneity of experimental units. While randomization helps create groups that are on average similar at the start of the experiment, homogeneity of experiments unit implies that the variability within each group is relatively consistent. This enhances the precision of each study and strengthens the ability to detect treatment's effect. Let's see the experimental setup of CRD. Experimental design overview. Plan, execute, and analyze experiments, defining objectives, select treatments, determining units, and the structure of the study. Random allocation of treatments. Randomly assign treatments for unbiased unit distribution, ensuring equal chances and enhancing the statistical validity. Factors and levels in CRD. CRD involves a single manipulated factor with different levels, that is, treatments, applied to units for experimental comparison. The ANOVA test and post hoc test. ANOVA test, statistical analysis to compare means of three or more groups to assess if there are significant differences. Determine if group variation is more than expected by chance. Post hoc test, additional analysis allowing ANOVA to pinpoint specific group differences when there's a significant overall effect, helping identify which groups are distinct. The statistical model in CRD. In CRD, a statistical model is utilized to analyze the relationship between the independent variable and the dependent variable. The dependent and independent variable are the outcome or response variable measured in the experiment. Meanwhile, the independent variable is the factor manipulated to observe its effect on the dependent variable. Formulation of hypothesis. Null hypothesis states no significant effect or difference among treatment groups. Alternative hypothesis posits a significant effect or difference resulting from the treatments. For example, in null hypothesis, there is no significant difference in the mean outcomes among treatment groups. Meanwhile, in alternative hypothesis, there is a significant difference in the mean outcomes among the treatment groups. Assumptions of CRD. Homogeneity of variances. It assumes that the variability in the response variable is consistent across all treatment groups. Homogeneity ensures that statistical tests accurately assess differences in mean without being influenced by unequal variances. Normality of residuals assumes that the residuals, the difference between observed and predicted values, follow normal distribution. Normality supports the validity of inferential statistic tests such as t-test and the analysis of variance, that is ANOVA. Independence of observations assumes that the observation within each treatment group are independent of each other. Independence is crucial for the validity of statistical inferences, and it prevents the influence of one observation on another. Compliance with these assumptions enhances the reliability and accuracy of statistical analysis in completely randomized design. Violation may affect the validity of results, and researchers often check and address this assumption during the data analysis. Let's see the limitations of CRD. Lack of blocking. CRD may not account for potential sources of variability that can be controlled through blocking. Blocking helps control known sources of variation, enhancing precision. Sensitivity to outliers. CRD can be sensitive to outliers, which may disproportionately influence the results. Other designs with built-in robustness to outliers, like randomized block design, might be more suitable. Here, the example is about the agricultural experiment. So, in the study, investigating the effect of three different fertilizers, treatment A, B, and C, on crop yield in a randomized field experiment. The analysis ANOVA revealed a significant difference in crop yield among treatments, that is, P is less than 0.05, and the post hoc test shows that the treatment B is significantly outperforming the others. So, in the interpretation, we know the treatment choice significantly impacted the crop yield, and the treatment B is the most effective one out of them all. Conclusion. In conclusion, understanding and adhering the principles of completely randomized design coupled with robust statistical analysis are vital for drawing valid conclusions. Proper designs and analysis enhance result reliability, ensuring meaningful contribution to scientific knowledge. Thank you for watching.